Hello, everyone, and welcome to today's live broadcast, Uncovering the Membrane Mechanism of Cytokinesis using Live Cell Imaging and Subcellular Optogenetic Tools. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Andor. For more information on our sponsor, please visit andor.com. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many of your questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top of your presentation window, or report your problem by using the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our speakers. Dr. Alan Mullan, Product Specialist for Microscopy Cameras at Andor. Dr. Jean Castillo, Postdoctoral Research Associate, Washington University School of Medicine in St. Louis, and Dr. Claudia Florindo, Product Specialist, Life Sciences, Andor. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Dr. Castillo, you may now begin your presentation.
Hello everybody, thank you for the introduction. My name is Jean Castillo. I will show the data from my work. The title is Uncovering the Membrane Mechanism of Cytokinesis using Live Cell Imaging and Subcellular Optogenetic Tools. Our lab uses optogenetic methods to spatially confine critical and molecular control in selected regions of a single cell. Dynamic networks of interacting signaling proteins govern cellular behavior in processes like cell migration, differentiation, secretion, and contraction. All those processes are controlled by signaling proteins that transduce the signal through the cell. The ability to selectively control the activity of specific signaling components in a network can be valuable in gaining a better understanding of the network dynamics that executes cell behavior. The most common used methods to study cell signaling are genetic methods like knockout, knockdowns, or the use of mutants, and pharmacological methods like the use of inhibitors, activators, agonists, or antagonists, but they have a limited control over location or time. Optogenetics gives us precise control over location and the time that we want to maintain the signal. Also, this technique let us see changes in real time. And like we mentioned, we have the control of the signal localization, then we can change the region or interest. Exist different optogenetic tools where you can optically activate membrane proteins like receptors and some G protein coupled receptors like opsins, or there are some constructs that let you activate receptor tyrosine kinases, RTKs receptors that let us activate the whole pathway, the whole signaling pathway, like when we use an agonist. There is other optogenetic tools are light sensitive and show conformational change in response to light. And some of these tools let us induce protein dimerization. With this, we can have optical control of the protein localization, recruiting proteins to different regions of the cell. And this construct let us activate or deactivate specific downstream proteins in the pathway. In red, we have the activated proteins, and in blue light we have deactivated proteins. The spectral selectivity of optogenetic tools allow us to image simultaneously fluorescent proteins or fluorophores while we are testing cell behavior. Using this approach, we deconstruct complex cell behavior to understand the mechanisms at their basis. Our optogenetic tools respond or is sensitive to blue light. This lets us image another fluorescent proteins like yellow proteins or red proteins at the same time. This is the microscope configuration that we have in the lab, where we have the mosaic and the mirror arrangement that let us perform localized photoactivation in a specific region of interest selected in the software. And also we have a dragonfly with where is the spinning disc and the turf and surf. In our lab, Patrick O'Neill developed an optogenetic tool that is based on light-induced dimerization. This tool let us achieve subcellular control over the activation of endogenous Rho A in a specific site of the cell. We optically triggered membrane recruitment of a Rho-selective guanine nucleotide exchange factor, GEF, for Rho A. The interaction between a protein SSPV and its peptide binding target SSRA is rendered light dependent by fusing SSRA to the blue light sensitive LOV domain. The LOV domain is attached to the membrane by a CAX motif, and this in the dark state is folded, hiding SSRA. Then when we shine light, the eyelid unfolds and lets the interaction between SSPV and SSRA. Then we recruit the GEF to the membrane that activates Rho A. I will show you some examples where we use this optogenetic tool, and also you can see one of the advantages that I mentioned before. We can change the signal localization and induce a different cell behavior. We work with macrophages, a cell line Rho cells 264-7, and we perform all the experiments in these cells. Also, like I mentioned before, we use a GEF, and this GEF is called large. It's specific for Rho A, and also it's known that Rho A mediates cell retraction. Then in the first movie, you will see an example of cell migration induced by Rho A photoactivation. In green, you will see the membrane marking, and also you will see a white rectangle or box that represents the region where we shine light. You will see this box in all my movies. And we can see how the cell moves in opposite direction to the light, exactly immediately when I start to shine light. In the second movie, we have the membrane marker in green. We are using a nuclear marker in red, and we can see like when I change and I photoactivate Rho A in the middle, the cell contract and form a cleavage furrow. The third movie, if I don't shine light, the cell does nothing. Once that we found that we are able to induce furrow formation by optical activation of Rho A, we decided to study the membrane mechanisms involved in this process that is part of cytokinesis, which is an important part of cell division. Then cytokinesis takes place at the end of anaphase and is the process to separate one cell in two. The process implies a cleavage furrow, then an intracellular bridge, and the last step is the pinch off or final cut at the midsome of the bridge. We think of in this process there is membrane remodeling, but how does membrane remodeling occur? To form the cytokinetic furrow and to create the intracellular bridge. Then our hypothesis is that spatially localized endocytosis and exocytosis play a role in this remodeling that is necessary for cytokinesis. This is based on the involvement of trafficking protein like RAF11 and RF6 in furrow formation. As we know, vesicular trafficking is the transport of the cell products between subcellular components like the endoplasmic reticulum and Golgi via vesicles to and from the plasma membrane and another specific cell location. There are two processes: endocytosis, that is the internalization of proteins and membrane from the plasma membrane to the cytosol, and exocytosis, the exportation of proteins and membrane from the cytosol to the plasma membrane. First of all, we evaluate in our system if the furrow formation was because the ROA activity was induced by our optogenetic construct. To check that, we use ROA activity sensor RGBD, the Rotekin GBD, which binds to ROA GTP, activated ROA form. Then we can look for activated ROA localization. In the movie, the red channel is our optogenetic construct, the green channel is our ROA activity sensor RGBD, and you will see like after furrow formation, the RGBD is localized in the furrow region. This means that the furrow formation is because we photoactivate ROA, and this is not an spontaneous event. Also, here we introduce a second box to form an intracellular bridge, and also I will tell you that we called this for my future movies. We call this region the back of the daughter cell and the front of the cell. Back, front. We know from a previous work in our lab that the activation of ROA induces actin myosin contractility that results in membrane flow. Then we check if there is membrane flow during furrow formation. For this experiment, we attach beads to the membrane. The beads are color red and the membrane marker is green. And like you will see in the first movie, after induced furrow formation, we see how the membrane starts to flow, the cell contract, and the beads flow to the middle, to the furrow region. Also in the green channel we can see membrane accumulation in the furrow region because the membrane flow. In the second movie we form an intracellular bridge after furrow formation, and like you will see, we found that the beads don't flow through the bridge. They stay in the back of the daughter cell. That means that the bridge is forming by addition of new membrane and not because mem flow. Like we suppose there is membrane incorporation by vesicular trafficking, and also it is known that low membrane tension induces endocytosis. Next, we use a low membrane tension marker, the forming binding protein 17, FBP17. And like you will see in the movie, the red channel is the membrane marker. In green, we have FBP17, which binds to the membrane with low tension. And after furrow formation, we see how the low tension marker is localized in the furrow region in the back of the daughter cell. Also in the red channel you can see some membrane accumulation because the membrane flow, and also this result tell us that there is low tension in the back of the daughter cells and endocytosis could be there. The next was track endocytic vesicles and their localization during furrow formation. To do this, we use a fluorescent ligand transferring Alexa 594 in red, and after 5 minutes of incubation, the transferrin Alexa internalized with the transferrin receptor via clathrin-mediated endocytosis. Like you will see in the first movie, after induced furrow formation, the vesicles containing transferrin Alexa in red localized in the furrow and in the back of the cell. The green is the membrane marker. In the second movie, we use another endocytic marker, cabelin, which is a membrane protein, one of the principal components of capillary membranes, and is involved in receptor-independent endocytosis. I mean, it's a different endocytosis mechanism, independent of clathrin. And in red is cabelin, the second movie, and the green is the membrane marker. And like you will see, cabelin localized in the furrow region, and if we keep photoactivating, it seems that is inside the intercellular bridge. That means that endocytosis is taking place, but all in all this region and in this process. Next, we look for another endocytic proteins, or we look for recycling endosome proteins, and in this case, we track RAV11. Like we know, the RAV proteins play an essential role in the process that underlie the targeting and fusion of transport vesicles with their appropriate receptor membranes. Then they are in charge of the sorting in endosomes. In the first movie, RAV11 is in red, the membrane marker is in green, and like you will see, after furrow formation, the RAV11 localized in the back of the daughter cell. In the second movie, when we knock down RAV11 by using a dominant negative protein in the green channel, the red channel in this case is our membrane marker, you will see that the cell can contract, but it cannot form a furrow. This means that the recycling endosome is important for furrow formation. vesicular trafficking is an important process during furrow formation. And if we inhibit a part of that, the cell cannot form the furrow. We look for exocytic proteins, and RF6 is a protein that is involved in exocytosis and endocytosis. And in the first movie, you will see in the red channel the membrane marker. In the green channel, we have RF6. And after induced furrow formation, we can see how RF6 localized in the furrow region, and also after some minutes it localized in the intercellular bridge. In the second movie, when we knock down RF6 by using a dominant negative in the green channel, and the red channel is our membrane marker, you will see like the cell contracts, but it cannot induce a furrow. It cannot form a furrow. And in the third movie, we use a constitutive active RF6, which was fused to the cyan fluorescent protein that we didn't image because technical problems, because if we can induce global photoactivation of our optogenetic construct if we image blue proteins. Then in the third movie, like you will see, the red channel is our optogenetic construct, the green channel is a membrane marker, and you will see that when we express RF6 constitutive active, the cell also cannot form the furrow. This means that the cell don't just need the RF6 activity, also it needs the dynamic switch on and off of RF6 during furrow formation. Another exocytic marker that we used was Exo70, which is part of the exocyst complex, and it is involved in the last step of exocytosis before membrane fusion. In the first movie, like you will see in the red channel, we have a membrane marker. In the green channel, we have Exo70. And after furrow formation, you will see like Exo70 is localized in the furrow and appears in the intercellular bridge. This makes sense because it's reported that Exo70 interacts with RF6 in exocytic process. Then we think that there is exocytosis in this region of the cell to probably form the bridge, and also it makes sense because all the machinery is recruited there or is localized there. In the second movie, also I will show how we can combine the optical control and use inhibitors, small molecules, and see the effect in real time. Then we use an inhibitor for Exo70, endocydin 2, and we'll see like after incubation we found that the cell can contract but cannot induce or produce the furrow, the cleavage furrow. Then that means that exocytosis is required for the furrow formation. Here I will present one of the most interesting results that I have, or one of the experiments that I like the most. And also this shows another advantage of the system configuration that we have. We can induce two different processes at the same time, endocytosis and cytokinesis, and then watch the effect with time and local control. Here I use the same fluorescent ligand, transferrin Alexa 594, to see more precise where endocytosis was taking place and what happens when we activate endocytosis while we induce furrow formation. Then the idea is to add the transferrin, induce the transferrin, then induce endocytosis of the transferrin receptor by desensitization and see what happened. And like you will see in the movie, in the red channel is the transferrin, the green channel is the member marker. Once that I form the furrow, I add the transferrin, and like you will see, in the same time that you start to see endocytic vesicles, the intercellular bridge start to elongate, and you can see some vesicles inside the intercellular bridge. Then enhancing endocytosis increases the bridge length. Then we ask what happens if we enhance the exocytosis. To do this, we overexpress cytohesin 2, a guanine nucleotide exchange factor for SRRF6. In the movie, the red channel is our optogenetic construct, the green channel is cytohesin 2, and we found that the overexpression of cytohesin 2 is enough to increase the bridge length. We found also that with this condition, we can form long bridges. This bridge is a little bit wider compared with the bridge when we enhance endocytosis. Then enhancing exocytosis increases the bridge length. And vesicular trafficking is required for the bridge formation. Looking for how similar was our optogenetic model, we look for some structural proteins that play a role in cytokinesis and also in the intercellular bridge formation. And we decide to look for septins. Septins are cytoskeletal proteins that form ring structures and are involved. These proteins are involved in cytokinesis, and they are involved in the intercellular bridge formation. Like you will see in the movie, the red channel is our member marker, and the green channel is septin 7. And you will see, also this is one of my longest videos, and you will see that when we overexpress this septin 7, we form we induce long bridges. We can increase the length, the bridge length. And also we keep photoactivating, like you will see. If we keep photoactivating, we found that these cells form a structure similar to the mid-body. Like I mentioned before, septins form a ring structure and also are involved in the mid-body formation. You will see, like after a while, seems that in the middle of the intercellular bridge start to appear this structure similar to the mid body. Then we think that septins localize in the intercellular bridge to confer or give this tubular structure and form the mid body. You will see here this structure. We think that the furrow elongation to form the intracellular bridge is mediated by membrane incorporation through exocytosis. And to see where exocytosis is, first we combine confocal microscopy, optical control, and fluorescence recovery after photobleaching, FRAP. And you will see in the first movie, in the red channel cabulin, in the green channel is the membrane marker, and I photobleach in the middle of the cell, and immediately I start to photoactivate to induce furrow formation. You will see that the fluorescence is recovered by membrane flow mostly, but you can see some vesicles going there. In the second movie, we induce, like we know that we are able to induce long bridges or long intracellular bridges by overexpressing cytohesin 2. We use this, and also we use a membrane marker, GPI, to see internal vesicles. And after induced furrow formation and form a bridge, you will see I photobleach in the middle of the intercellular bridge. You will see the box there, and you will see how some vesicles move inside the bridge. There is another photobleaching cycle, and you see how the fluorescence in the membrane is recovered, and it seems that some vesicles fuse with the membrane. Then this means that there is exocytosis in the intercellular bridge. Next, we use total internal reflection fluorescence, TIRF, which is ideal for visualizing events such as exocytosis, which occurs at or near the plasma membrane around 100 nanometers above the substrate. And we combine TIRF and optical control to see more clear exocytosis, and we use BAM2, a protein involved in membrane fusion. BAM2 is fused to a pH-sensitive protein, which is less bright in endosomes, in acid pHs, and it is brighter when it's in the membrane, pH 7. And you will see in the movie, in the left channel, the black line represents the outline of the cell, and you will see BAM2, and after furrow formation we can see how some dark spots appear in the front of the cell. You see here. Then we can see that BAMP is polarized in the front, and these results show that there is exocytosis at the front of the cell. Also, BAMP stay in the membrane for a while. The kinetics of BAMP is around 20 minutes before comeback in endosomes. To see independent exocytosis event, we use again TIRF combined with optical control of the furrow formation. And in this experiment, we use the neuropeptide Y, which has been used like a secretion marker. The MPY is also fused to a pH-sensitive protein, pH tomato, which is less bright in endosome acid pHs, and it is brighter when it is in the membrane, pH 7. Also, MPY is secreted to the extracellular milieu and diffuse. Then you will see in the movie the left channel, the outline represent the cell, and after induced furrow formation, we found how some dark spots appear and disappear in the front of the cell there, like you can see. Then this is another result that tells us that there is exocytosis at the front of the cell. To see if the cell is using exocytosis to incorporate new membrane and to see from where this membrane comes, we perform the next experiment. We use a fluorophore to stain the membrane FM464, which is red. Also, it can be washable from the plasma membrane. Then if we look the scheme, I incubate the cell with FM464, and after 30 minutes, it gets internalized and it stains internal vesicles. Then I wash it from the membrane, keeping just the vesicles containing FM464. If the new incorporated membrane comes from endosomes, then the plasma membrane will stain again. For this experiment, I combine surface stream, turf imaging, and optogenetic. In the movie, like you will see, the first channel is FM464. In green is we have clathrin, which we use like a control, just to check that the cell is not just getting close to the surface. And like you will see in the beginning, the vesicles are in the middle. After induced furrow formation, we can see some vesicles moving to the front, and after some minutes, a line of FM464 fluorescence appear at the front edge of the cell, indicating that vesicles are incorporated into the plasma membrane. Also, you will see some here, like some arrows that show this. And this is in contrast to clathrin, which appear in vesicles but didn't form a node line at the front edge. Then also we could see just this membrane incorporation by using surface stream imaging. You can see here the line. Then we propose a model where ROA activation in the middle of the cell causes ingration and cleavage furrow formation due to the actin-myosin contraction and plasma membrane flow to the site of ROA activation. This leads to decreased membrane tension in the furrow and increased endocytosis. As the intercellular bridge forms, endocytosis is localized to the backs of the nascent daughter cells. The endocytosed vesicles undergo exocytosis in two distinct regions using two different mechanisms. In the first mechanism, the vesicles are trafficking to recycling endosome containing RAF11 and then to the intracellular bridge where they undergo RF6 and Exo70 mediated exocytosis. This supplies membrane lipids needed for the bridge formation. In the second mechanism, the vesicles undergo exocytosis at the front of the daughter cells mediated by snare proteins like BAM2. This mechanism serves to replace the plasma membrane lipids at the front edge, allowing the daughter cells to maintain their shape as they move apart and elongate the bridge. Then, as we see, subcellular optogenetics provides experimental control over complex cell behavior so that the mechanisms can be addressed. Here, we use this approach to show that localized trafficking controls cytokinesis, and also this technique can be used similar to understand the basis of other cell behaviors, and it can also be used in the future for therapeutic purposes. Thank you so much. Thanks to my lab, especially Sinia Meshik and Anu Pandi that helped me a lot in this work. Thank you.
In this part of the presentation, we're going to look at SurfStream, which is a cell-friendly, flexible, and accessible super resolution method. So we're going to look first of all at what SURF is and how it compares with a number of other super resolution methodologies, and then also looking then about what is SurfStream and how this differs from normal SURF, and then how you actually can use SurfStream for live cell imaging purposes. So as we know, super resolution techniques are used to see beyond the diffraction limit, and conventional light microscopy methods, of course, are limited by diffraction, and that works out to be around 200 to 300 nanometers. And below that range, the inner workings of the cell are essentially lost in that haze. But we can consider information beyond the diffraction limit in terms of the point spread function, and of course now we have a wide range of super resolution techniques that allow us to view subcellular structures and processes in detail. I'll not go into these and the workings of these in any detail, but obviously we have a range of them from STORM, PALM, STED, and also SIM as well. And each of these will have their own strengths and disadvantages. So some of the limitations include having to use specialized equipment such as high power lasers or particular optical setups. Also, we may need to use special photo switching fluorophores, so-called blinking fluorophores. But the other one that of most concern to us for live cell imaging really is in terms of illumination. And so illumination intensity is particularly important for live cell imaging. We can see that from a technique like wide field, we might be in order of milliwatts per centimeter squared, SIM moving up into watts per centimeter squared, and then into PALM and STORM where you are dealing with into the kilowatts per centimeter squared, which is really quite a high level of light. So this brings us to SURF, or to give it its full title, Super Resolution Radio Fluctuations. And this was originally developed by Ricardo Henriquez and his colleagues back in 2016, so it's been around for a few years now. And what this does is it provides a cell-friendly, flexible and accessible super resolution technique. By cell-friendly, we mean that this is an approach which is analytical based and requires orders of magnitude less light than the existing methods that we've just mentioned. And it's flexible and accessible to all, so it's purely a software-based super resolution approach. There's no need for any specialized equipment, and also it is able to use any fluorophores, so standard GFP or other common fluorophores allow us to have flexibility over the labeling that we're going to use. It can also use any imaging modality, so that can be wide field, TIRF or Confocal. And the post-processing version of SURF is a free and open source ImageJ plugin. So how does SURF work? Well, what we do with SRRF is we capture a burst of image frames of very short exposure with the view to trying to localize the emission from a point source of a fluorophore. And then we upscale the pixels of the camera into virtual pixels essentially. So this allows us to localize the emission more accurately. And then using a GPU, we're able to compute the local intensity gradients from each of these pixels, and then we can interpolate the gradients then in this virtual array of subpixels, and that allows us to quite precisely and accurately work out where the original source of the illumination is. And key to this is having the GPU. And of course GPUs are well known for other techniques now, such as for deconvolution. And by using the power of this sheer processing of the GPUs, we're able to do this very, very quickly. And so we're taking 100 frames, very short exposures, say 10 milliseconds, and then we're trying to work out where the location of the emission is from. And then we can rebuild the image from that, and then we have this super resolved image. Okay, so we've just mentioned what SRRF is. So what then is the difference between normal SRRF and SRRF stream? Well, SRRF stream is a development of SRRF which is exclusive to Andor technology, and this came about because of a collaboration with Ricardo Henriquez, and that Andor had a lot of experience of GPU optimization for deconvolution. So what we did then is that we were able to optimize the GPU operations and greatly enhance the speed of SRRF. And what that allows is for actually real-time integration of the acquisition events and the SRRF analysis. And that provides real-time super resolution, so it is not just the normal post-processing of the normal SRRF. So how can you use SRRF stream for live cell imaging applications? Well, one option, as we're going to hear about later, is to do with using it with the DRAGONFLY Confocal system. Then another option is basically any microscope. You just need to have an Andor Exxon EMCCD camera, which provides the sensitivity required to operate at these very short exposures. Then a SRRF license that is locked to the camera, and then of course the GPU enabled PC. So what resolution and speeds are possible from SRRF stream? Resolution wise, typically 2 to 6 times better than the original data is possible. So that's using a high NA objective and ensuring that the image itself is properly sampled according to Nyquist criteria. So that means that 50 to 150 nanometers of lateral resolution is possible depending on the sample and the fluorophore density. So a low density of fluorophore will tend towards giving a higher resolution. Likewise, taking more samples will provide improved resolution as well at the expense of lower speed. And speed wise, with a normal configuration of taking 100 frames, each with 10 milliseconds exposure, that is equating to around 1 frame per second. But of course we can start to look at regions of interest, and as well as using a slightly smaller number of input frames, and then that way that we can start to boost speeds up higher between 2.5, 5 and over 10 frames per second. So in summary, SRRF stream is an Andor exclusive version of SRRF that exploits optimized GPU processing. It shares many of the key benefits of SRRF and adds in a real-time dimension to this. So that provides a real-time live cell super resolution solution from a camera with a standard microscope setup, or alternatively via the Andor DRAGONFLY Confocal system. So it will run in Micromanager in a camera-based setup or within the DRAGONFLY via Fusion. And SRRF is highly applicable to analysis of subcellular organelles, tracking and membrane studies, as we've just heard, primarily because of this, the low illumination and the high speed live cell aspects of it, coupled with the conventional fluorophore side of things. So now we are going to go on to hear more about the Andor DRAGONFLY.
Thank you, Dr. Castillo and Dr. Mullan, and thank you, Christy, for handing over to me now. I will wrap up showing Andor solutions for imaging and photo manipulation. So I just would like to give a first start of the imaging challenges that Dr. Castillo faced when was designing his experiments. So he performed, as you've seen, a lot of live imaging, and when a researcher does a lot of live imaging, there are challenges that are very obvious when performing live imaging, such as phototoxicity and photobleaching. And also in the case of Dr. Castillo, speed was a very important requirement, the high temporal resolution that he needed to induce photo manipulation and acquire the images. You have also seen Dr. Castillo delivering multiple imaging techniques, such as Confocal, TURF, SRRF, and FRAP. He also performed super resolution, and there are several challenges when facing live cell super resolution. He used optogenetic tools to induce the cleavage furrow, and then he needed an imaging system that would acquire with high temporal resolution the induced furrow. So I will now point out Andor solutions to address these imaging challenges. Andor has the DRAGONFLY multi-point spinning disk based on the microlens spinning disk technology. DRAGONFLY is a multimodal platform. It is an instant multi-point confocal that also delivers laser wide field imaging, TURF, super resolution modalities, D-Storm and SRRF, with a very broad range of excitation wavelengths from the visible to the near infrared, and a very flat illumination across the whole field of view, the Borealis patent Andor illumination system. You can see the comparison between the DRAGONFLY and the single-point scanning system, where the images in a multi-point confocal are delivered much faster than in a single-point scanning confocal. And with faster acquisition, you increase the temporal resolution of your images. Because we have a dual spinning lens system, there's higher efficiency in the capture of light, and therefore it's less phototoxic. And this, combined with Andor's high-sensitive detectors just presented by Alan, deliver the capture of more photons of an image, and it's less phototoxic and less photobleaching, and you can capture even the dimmest signals in a cell. The specifications of the DRAGONFLY include acquisition speeds up to 400 frames per second, in the confocal mode, a very large field of view to match the 5.5 megapixels Zyla camera within a 16-bit dynamic range of acquisition, a broad range of excitation wavelengths from the 405 till the near infrared wavelengths, 785 nanometers, and a highly uniformity across a broad range of wavelengths and across the whole field of view. And due to all of these characteristics, it's not surprising that the DRAGONFLY has a broad range of applications for life sciences, such as live and fixed cell imaging, membrane dynamics, example exocytosis, developmental biology, cancer research, organoids, neuroscience, and single molecule localization imaging. I now propose you to analyze the live cell super resolution challenges. Techniques that are compatible with live cell imaging are wide field and confocal imaging, multi-point spinning disk confocal, but when you want to break the diffraction limit of light, many of the techniques available are not compatible with live cell imaging, moving into the red and red areas, being less and less compatible with live cell imaging. And this is due to high laser power requirements, also to the acquisition timings that can be from minutes to hours, and therefore you have a problem in the delivering the temporal resolution needed for live cell imaging in many experiments, and also very complex probe preparation. In 2016, Professor Ricardo Henriques have developed a super resolution technique that is compatible with live cell imaging that is named SRRF. Professor Ricardo Henriques further collaborated with Andor to develop a GPU-based algorithm embedded into our cameras and into Andor DRAGONFLY software, which is Fusion. SRRF yields an increase in resolution between 2 to 6 times more than conventional diffraction limited data, giving resolutions from 50 to 150 nanometer final resolution. The low power requirements that are needed to deliver a SRRF image make it compatible with live cell image. SRRF algorithm Andor's SRRF algorithm allows acquisition of images that break the diffraction limit of light as fast as 10 frames per second. And importantly, SRRF images can be acquired deep inside cells or tissues. Another characteristic that I would like to show you of the DRAGONFLY is that due to the Borealis uniform illumination and with the stitching algorithm embedded into Fusion, you can acquire large organisms and stitch it with seamless stitching, having very detailed resolution at very high magnifications, but at the same time being able to visualize all the organisms that you wanted to acquire. So the DRAGONFLY is offered in two models, the DRAGONFLY 500, a complete solution with all the imaging modalities that the DRAGONFLY has to offer, and the DRAGONFLY 200, a more compact solution that is offered in the upright and inverted systems. And I'm now going to talk about the photomanipulation tools that Andor has to offer. Andor offers the micropoint and mosaic as the photomanipulation devices. Mosaic delivers Andor patent DMD technology. It is composed of 500,000 individual mirrors. Those mirrors are controlled individually through the software, and this delivers a very precise control over light in both space and time, being able to illuminate different regions simultaneously. And this illumination devices and controlling the different mirrors simultaneously allows the illumination of different regions of a cellular structure at the same time. In this movie presented, you can see multiple regions being illuminated simultaneously with the mosaic. Micropoint provides a flexible and field-proven tool for photostimulation. It is delivered with Andor patented compact pulse laser nitrogen pumped tunable dye laser, and it's capable of exciting and photoactivated from a wide range of wavelengths from the 365 to the 656 nanometers. Micropoint can be used in several applications such as ablation, bleaching, and uncaging. And in this video, you can see the photoactivation of vesicles turning from green to red using the micropoint. For this photoactivation, vesicle distance, track, speed can be determined with Imarish software. So I will wrap up presenting why Andor could deliver all the imaging challenges faced by Dr. Castillo. Dr. Castillo needed a live imaging system that would be able to deliver images that could overcome the challenges of live cell imaging, such as phototoxicity and photobleaching, but could also do so acquiring with a very high temporal resolution. The Dragonfly delivers images with very low phototoxicity and photobleaching due to the combination of the dual microlens system that is highly efficient in capturing light and of Andor's high sensitive detectors, EMCCD and SCMOS detectors, meaning that you can acquire images with high quality when even using very low light exposure. Dr. Castillo used multiple imaging techniques, Confocal, TURF to analyze membrane uptake at the cleavage furrow, SRRF to image very high dynamic small details, and FRAP to induce photoactivation of the regions at the membranes. And to do all of these imaging techniques, either you have multiple imaging equipments or you have a system, the only system that could deliver all this imaging modality in a single row, which is the Dragonfly. Super resolution in live cell imagings was also delivered due to Andor surfstream GPS algorithm that is used and appropriate for live cell super resolution. Combining the optogenetic devices with the higher temporal resolution of the Dragonfly, Dr. Castillo was able to induce the cleavage furrow with the optogenetics and then to acquire immediately after the induction of the cleavage furrow, and therefore to deliver a solution for how cytokinesis membrane is remodeled in the pre-cytokinesis abscission. And this is all for now. Thank you so much for listening.
Thank you, Dr. Florindo, Dr. Castillo, and Dr. Mullan for your informative presentations. We will now start the Q&A portion of our webinar. If you have any questions you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have so many great questions already coming in. Let's start with this one. Dr. Florindo, the Dragonfly is a spinning disc. I had the impression that the spinning discs are suitable for live imaging in thin samples, but not that good for thick sample imaging. Could you comment on this?
So thank you so much for this excellent question. Traditionally, multi-point confocal systems, spinning discs, they were not very good for imaging 6 samples due to crosstalk on the pinholes. And in these older systems, the crosstalk would actually begin in specimens above the 5 or 10 micron depth. But in Dragonfly, the 40 and 25 microns pinhole provided, and with Andor custom pitched pinhole spacing, results in 10 times more background rejection than traditional spinning disc systems. Also, when comparing single pinhole confocals with point scanner systems with spinning disc systems, point scanners show excellent performance in background rejection, but in practice, comparing with the Dragonfly using real samples, we have delivered surprising results. The slow scanning speed and high bleaching rates of the point scanners, combined with inferior sensitivity, result in surprisingly poor imaging performance in thicker specimens. We actually have clients who have abandoned their projects on 6 samples that could not be done in point scanners due to bleaching or slow speeds. And those projects were actually gained live again with the Dragonfly. And I must say that we have clients that were able to image samples within millimeters of depth in clear tissue. So nowadays, and with the DRAGONFLY, you can image samples that are sick as well as samples that are thin. Thank you so much.
And thank you, Dr. Florindo. Dr. Castillo, let's come over to you for this question. If you activate row A and add a row inhibitor, will the furrow still form?
Thank you so much. This is a curious question. Usually, in these cells that we perform the experiments, the row cells, the macrophages, they express three isoforms of row, A, B, and C. And the most isoform that they express is row A. Another thing is that the gap that we are using to activate row A is specific or has more affinity for row A than the other two isoforms. Then I think it's one of the things that we didn't try. And now that it would be curious to see and interesting to see if once that you inhibit row A and you is still recruiting this gap, it would be really interesting to see if the other two isoforms can substitute the row A activity, and that is enough to induce furrow formation, or just those proteins are not required for this, and it's more like a specific row A. That is an interesting question because it's something that we didn't try, but basically we didn't try because we were sure that the gap that we are using is more specific for row A.
Thank you, Dr. Castillo. Dr. Florindo, let's come back over to you. And this is a few parts here. From the photo manipulation tools presented, I would like to ask, one, can they be combined with the DRAGONFLY? And two, what are the main differences between mosaic and micropoint?
So as just presented by Dr. Castillo talk, the photo manipulation tools presented can in fact be combined with the DRAGONFLY. And I will now pass to the second part of the talk, the question, which photo manipulation tools Andor offers. So Andor offers the mosaic and micropoint. And the main differences between the mosaic and micropoint are that micropoint allows single point illumination, and you can use it for higher laser power applications, such as ablation and DNA damage. Whereas mosaic is a device more suitable for optogenetics, is extremely fast, and allows simultaneous multi-point illuminations of complex regions. Thank you so much for your question.
And thank you. Dr. Castillo, another one for you. This one has two parts. Have you measured membrane traffic across the midbody? And if not, could you comment on what you think is happening in the midbody pre-abscission?
Okay. One of the things that we did in this work, and like we studied the trafficking in the intercellular bridge. We didn't quantify. We just we saw we photo bleached the intercellular bridge, and as we saw in the presentation, we could track some vesicles going through the intercellular bridge that let us hypothesize that the exocytosis is happening there. About the mid-body and the final cut, one of the things is we think that this process is more complex and requires another signal and the recruitment of other proteins machinery that takes to the final cut to split the cells. completely into independent cells. Thank you.
And thank you, Dr. Castillo. And we have time for one more question. Dr. Florindo, let's have you answer this question. What are the super resolution options offered by the Dragonfly?
So thank you so much for this question. And I will now talk about the super resolution options offered by the Dragonfly. So the Dragonfly offers D-Storm and SurfStream. And I will explain a bit more about what Andor offers in each of these super resolution modalities. In the Dragonfly for D-Storm, the same laser that is used for confocal imaging, wide field, and turf can also be applied for D-Storm. And to achieve the high power laser requirements for D-Storm, Dragonfly Borealis illumination system is designed with 4 motorized illumination zooms, which will increase the power density of the incident light up to 36 more times. We can deliver 3D single molecule data with D-Storm by the use of an astigmatic lens that will create a distorted PSF that varies with the actual diffoxes. Researchers will actually be able to determine the 3D single molecule localization data of their molecule of their interest. Resolutions up to 30 nanometers are achievable with this storm. We also provide, as very well presented by Dr. Castillo in the talk, SurfStream. And SurfStream is embedded into Andor's cameras and Fusion, the Dragonfly software. And it's a GPU-accelerated algorithm. So SurfStream implementation of Dr. Enrique's Surf algorithm allows you to acquire images in real time, super resolved images. And due to its low power requirements, it's compatible with live cell imaging. It can be combined with any imaging modality offered by the Dragonfly, wide field, confocal, or turf. It is extremely fast, delivering super resolved images with speeds up to 10 frames per second, and it can deliver super resolution images deep inside the cells and tissues. Very importantly as well, SurfStream images do not require any specific sample preparation. Thank you so much.
Thank you, Dr. Florindo. Now, I would like to thank our audience for their time today and for their interesting questions. Those questions we were unable to answer live today, and those that are submitted during the on-demand period will be addressed by our speakers via the email address you provided at the time of registration. I would once again like to thank Dr. Claudia Florindo, Dr. Alan Mullan, and Dr. Jean Castillo for their time today and for their important research. We'd also like to thank LabRoots and our sponsor Andor for underwriting today's educational webcast. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Have a great day.